The cell cycle is divided into two phases, interphase and cell division. Next, you will see a short video showing a cell dividing. Afterwards, we will look at each step in the process. The cell cycle begins with interphase. The cell has just divided and needs time to grow to normal size and synthesize all of the organelles needed for the cell to survive. Interphase consists of three parts. During G1, the cell is growing and making necessary parts. Also during this time, the cell is doing its job. All of the functions necessary to keep the organism alive, such as protein synthesis, are happening during G1. During S phase of interphase, each chromosome makes an exact copy of itself. G2 is the last part of interphase. During this time, the cell gets ready to divide. This picture shows a single chromosome on the left and the duplicated chromosome on the right. These two identical chromosomes are called sister chromatids and are held together by the centromere. Because we cannot see individual chromosomes with a microscope during interphase, most pictures that we see of chromosomes look like this one, with two sister chromatids. Cell division is divided into two parts, division of the nucleus, which is called mitosis, and division of the cytoplasm, which is called cytokinesis. The first step in mitosis is prophase. During this time, the chromatids condense and thicken, the nuclear membrane and nucleolus disappear, and the centrioles move to opposite ends of the cell, forming spindle fibers between them as they move. In the cell showing early prophase, we can still see remnants of the nuclear membrane, and we can't see the individual chromatids yet. In the late prophase cell, the nucleus is gone and the chromatids are clearly visible. The second step of mitosis is metaphase. During this phase, the spindle fibers attach to the centromere of each sister chromatid and move them to the center of the cell. Meta means middle. In this picture, we can clearly see the chromatids lining up in the center of the cell. Anaphase is the third step in mitosis. In this phase, the spindle fibers pull the sister chromatids apart and drag them to opposite ends of the cell. Once the sister chromatids are separated, they are now called chromosomes. Because they move to opposite ends of the cell, this ensures that each new cell will contain identical genetic information. The fourth and last step of mitosis is telophase. It is the opposite of prophase. The spindle fibers break down and disappear, the nuclear membrane and nucleolus reform, and the chromosomes relax and lengthen. In this picture, you could see two nuclei in one cell. Towards the ends of telophase, cytokinesis begins. The cell is divided into two cells that contain identical nuclei. The next video takes you through the steps of the cell cycle. See if you can identify each step as it happens.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.